This is the third part of today's session on EEG. After we know how the basic principles of EEG recordings in the lab work, we can have a look at a very specific analysis method that is widespread in psychological research, the event-related potential, or ERP. So here you can see a participant sitting in our lab looking at the screen and pressing a button according to what they see on the screen. So you can see this is some kind of working memory task. You don't really need to understand this now. We will get into this in the working memory session. But what is important to note right now is, as you can see, the participant is wearing a EEG cap. So we're interested, obviously, in what's happening in the brain while the participant is doing this task. And how do we do this? Well, we need to link the EEG signal to the events that are of interest to us, namely the appearances of stimuli on the screen. So if you think of the EEG signal as a continuous stream of data, then the challenge is really to know which part of the EEG belongs to which part of the experiment. So we do this by sending markers to the EEG amplifier. So for example, there might be a marker for the trial onset, there might be a marker for the stimulus presentation, and also for the response. So throughout one trial, there might be several markers coding different things, different events, and then the marker informing us about the experiment is stored together with the spontaneous EEG in a data file. So what we typically have is different markers for different conditions. As experimental psychologists, we're of course interested in comparing different conditions and seeing how an experimental manipulation affects our EEG signal. So imagine our participant here is seeing different faces on the screen, and we want to compare how the brain responds to different kinds of faces. So for example, here you can see happy and angry faces. So how do we do this? Here you can see a couple of electrodes. We would normally do this for at least 32 electrodes, but this is just an example. And then what we do is we look at some time window after the onset of each stimulus. So for example, here you can see this is a neutral phase, this is an angry phase, and this is a happy phase. Whenever you see the neutral phase, there will be a marker stim3. Whenever there's an angry phase, there will be a marker stim1. And then we have this segment of EEG data, which is typically, I would say, between 500 milliseconds and maybe 2 seconds. It depends really on the process, on the cognitive process you're interested in. In attention, something like 500, 700 milliseconds is typical. And then we actually cut out these segments, so we're not interested in these parts between the data, between the stimuli where nothing is happening on the screen. We're really just interested in, say, the 500 milliseconds after the stimulus onset, and maybe also the 200 milliseconds before the stimulus onset. So what we do then as a next step is we sort these segments according to the condition. So for example, if we're only interested in angry and happy, we can sort these. So each row here is an individual trial, and then you can basically sort them into these two conditions, and then you have a bunch of trials that belong to each condition. One thing you might notice here is that they seem to be a bit misaligned. So this one is somehow above this x-axis, this one is below it, and so on. So in order to make them more comparable, we do something called baseline correction. So the principle of baseline correction is there is constant variation in the EEG data, and we assume that, say, the 200 millisecond epoch before each stimulus is some kind of baseline. That's like the so to say, the standard EEG level at which we are expecting EEG the EEG signal to be at, And then anything after these 200 milliseconds due to the stimulus onset would be the actual deflection in the EEG signal due to the onset. So how do we correct for any variation in this 200 millisecond interval before the stimulus onset? Well, that's very easy mathematically. We want to shift the waveforms towards zero. So what we need to do is we need to calculate for each trial the average activity before zero. For example, 200 milliseconds before. You can also use 100 or 300. It depends a little bit on how densely the stimuli are presented. But let's just say 200 milliseconds before the stimulus. We take the average of that and then we subtract that average from the entire wave. And then what happens is that they are shifted towards zero. If we did this now again and took the average of the 200 milliseconds before, this would be zero. And here, of course, it would not necessarily be zero, but now we have a representative waveform here. And that's important because we want to compare the conditions and also compare between participants. The next step is the so-called artifact rejection. So, as I said, we want to try to avoid any unnecessary deflection in the EEG data that is not due to our experimental manipulation. We call noise anything in the data that is not related to our experimental manipulation, and we want to avoid that in the first place, for example, by telling participants not to chew chewing gum, not to blink when there's something presented on the screen. We use active electrodes to increase the signal-to-noise ratio, but there's always some remaining artifacts. So artifacts is an artifact is an EEG data. Is a part of the EEG data that is contaminated with noise. And what we need to do then is to take these trials, take each trial, look if there is an artifact present or not, and if there is one present, then we will reject that trial and not use it for further analyses. So examples for artifacts are, for example, so these trials would be okay, there doesn't seem to be any noise here, but then saccades would be something like this. You can see this typical step function. So here a person is looking to the left and then to the right. Here it's just to the right. And this is problematic because at this point we can't really use the EEG data. We don't know what's going on in the brain. This is really just an artifact induced by the eyes being a dipole. So we would get rid of these. This is a drift. So this might be, for example, a participant sweating. It could also be that an electrode is not perfectly attached to the cap, or that the dry gel is preventing a good electrical connection. Here we have a blink. That's another thing we would need to extract. And this is some random noise. So this could be that the electrode is completely detached from the cap, or it could be a faulty electrode. So we need to take these out and just keep those that we think do not contain any artifacts. Then the next step is averaging. So for each condition and electrode, we need to separately average across all the trials that are still there after the artifact rejection. So for example here, this is for PO7 for one electrode on the left hemisphere, and then FPZ would be a central frontal electrode. So for each of these electrodes, we average across all the trials that we have for condition 1, condition 2, same here. And of course we would do this for all electrodes. And why is it so important to have many trials? So in EEG experiments, other than just behavioral experiments, we need many more trials. Sometimes it depends a little bit on the ERP component of interest, but we might need up to 200 trials per condition per cell in our in our experimental design because of the properties of that ERP component. So why do we need so many trials in EEG typically? Well, the reason is that there is still a lot of noise in the signal that we measure. And this is best described by the additive noise model here that you can see. So the signal on the right hand side here, this is really what we observe. So this is the input to our recording computer. And then this is more of a theoretical understanding of the signal that we're measuring. So here we have the actual evoked potential. So something is happening, and then we see this typical waveform as a response to that signal. And then we have noise. So noise is always defined as a random signal. Random meaning it's not related to our experimental manipulation. It could be anything from different thoughts the participant might have. In one trial they might be thinking of a hot cup of coffee. The other trial they might be thinking of their last vacation in Portugal. So there's always some randomness there. It could also be, for example, a random movement of your head. It could be anything related to electric noise, for example, an electric engine from an escalator nearby. So all this noise we don't really know anything about. To us it is random, but it will still appear in the signal, of course. So what happens if we don't just take a single trial like this? This is a single trial, but say we average across 20 trials. What you can see is that the noise is reduced, whereas the evoked potential, so the deviation in the EEG waveform that we are interested in, remains the same. And then the signal also seems a little less noisy, and maybe we can even recognize the evoked potential in the signal a little bit. Why is the noise reduced? As I said, the noise is random, so it's not related to our event, in our event-related potential. So because it is not related to that event, if we average across 20 trials, then it should average out at some point. It should at least be reduced for 20 trials, because the signal is unrelated to the event. So in each trial it will look different, and then if we average across 20 trials it will be closer to zero. And if we don't do this just 20 times but 100 times, we can see that the noise is even further reduced, because now there's even more of a chance for the noise to cancel each other out. But the evoked potential is of course the same, because that is not random but related to our event. And then what we see in our measured signal is that we can now see maybe a more typical ERP pattern here. Still some noise, but this is already pretty good. Okay, and then what we see after averaging is this event-related potential here, and we can compare, for example, our conditions. Condition 1 and condition 2, here denoted as a blue and red line. And the interesting thing is now that we have distinct components in the ERP, for example, P1, N1, P2, N2, P3. There's others. And we can use these ERP components to target very specific cognitive functions. So this allows us, for example, to see where in the EEG or where in the ERP we see a difference between the conditions, and this allows us to then conclude which cognitive process is affected by our experimental manipulation. So P1 means it's the first positive deflection in the EEG signal, N1 is the first negative one, P2 is the second positive one, and so on. Please note that negative is always plotted upward in EEG world. So what we can see in this example is that there's no difference between the conditions in P1, N1 and P2, but there is probably a difference in N2 and P3. Of course, this would have to be statistically evaluated, but just by face value, that's something I would conclude first. So in order to do these statistics and actually evaluate whether there is a meaningful difference between conditions, we need to export the data. So we need to calculate the mean amplitude for all conditions and all electrodes in a certain time window of interest and then through statistics. So for example, for subject 3, 11 and 16, just to show a few examples, you can see a similar pattern. So blue is always more negative in this area than red. So we would take the average for blue and red for this subject, for blue and red for this subject, for blue and red for this subject, and then we might end up, if there's only 2 conditions, we would run a t-test, I guess. But this can be, of any complexity. So, just to highlight this once more, the statistics are of course done on the event-related potential for each subject, and the grand average, which is across all subjects, is really just for display purposes, so that the reader or someone looking at the data can get an idea of like the typical response across all participants in that specific task. You can also show event-related potentials as topographical maps. So if we have a grand average like this, and for a certain electrode CZ here, we can, for example, show the average difference between blue and red in this time window and in this time window, and then display it on this head. And then if we don't just do this for ZZ, but for all electrodes, and then we color code the amplitude, we will get a color map like this. So this is really not any other data than we would have here. It's just a different way of displaying it. You get a better idea of the distribution of components across the scalp. So while here amplitude of the EEG signal in microvolt is shown as a certain value on the y-axis, of course, here it is shown as a color. So I pointed out several times that it's very important to obtain good data quality, good signal-to-noise ratio. It is already quite demanding of participants to let them do so many trials. So we should do any effort possible to to reduce the noise and increase the signal-to-noise ratio. So increasing the number of trials is always an option, but of course you have to think of the participant as well. You don't want them to feel too uncomfortable. There's also effects of fatigue and not paying attention to the task as much anymore. EEG experiments can last up to 3 or 4 hours with breaks, of course, but still that is very demanding of the participant, and increasing the number of trials is not always feasible. Decreasing the impedance is very important, so the electrical resistance between the skin, the scalp, and the electrode should be reduced with the gel, and sometimes you need to monitor that. I mean, you always need to monitor that during the experiment, and then sometimes you need to readjust and add maybe more gel, for example. Generally, it is advisable to avoid any noise during recording, so use a Faraday cage, low temperature, so that participants are not sweating. Instruct them to not move around during the task. In the breaks, that's fine, but not during the task. Do not move the eyes while there's something on the screen that you're interested in getting a response of in the EEG signal, blinks, etc. And then active electrodes is also a good way of reducing noise. So we have active electrodes here in our lab in Southampton. They are more pricey than passive electrodes, way more, but from a scientific perspective, they're definitely better. So here are a few examples of ERP components that are associated with certain cognitive functions. The P1N1 is related to sensory gain and early attentional processes. The N170 to the detection and processing of faces. N2PC is a lateralized component. We will go into detail about that later. Related to attention. P3 is related to the evaluation of and categorization of stimuli. The N400 is related to semantic processing, and the LRP to the preparation of motor responses. So this is just giving you an overview of how different cognitive functions are reflected in the ERP depending on what latency you're looking at, and also these components. It's not really shown here, but these components are most prominent in different areas of the scalp. Okay, so much about how event-related potentials are calculated. In the next part, we will talk about how they are actually used in psychological experiments, and I will show you some examples.